There are 5 classes of antibodies or immunoglobulins, named as Immunoglobulin G or IgG, Immunoglobulin M or IgM, Immunoglobulin A or IgA, Immunoglobulin D or IgD, and Immunoglobulin E or IgE. All these classes have the basic 4-chain antibody structure. Also, they all have 2 types of light chains named as kappa and lambda. Both types are very similar in structure and no functional difference as well, but are coded for by different genes. Difference between these classes comes in heavy chain that are different for each class of immunoglobulins. They are termed as gamma for IgG, mu for IgM, alpha for IgA, delta for IgD, and epsilon for IgE. The differences among antibody classes are most pronounced in the Fc regions of the antibody classes, and this leads to the triggering of different effector functions on binding to antigen. For example, IgM recognition of antigen might lead to complement activation, whereas Ig recognition, possibly of the same antigen, might lead to mast cell degranulation and anaphylaxis. Structural differences are also very pronounced among different classes of antibodies. The major antibody in the serum is IgG, and as this is the best understood antibody in terms of structure and function, so we shall consider it first. The other antibody classes will be considered in relation to IgG. So let's look at different classes of antibodies one by one. Starting with immunoglobulin G. This is the most abundant antibody in serum, as it constitutes approximately 75% of total serum immunoglobulins. It is the simplest of all antibodies in terms of structure. It consists of similar 2 light chains and 2 heavy chains. The light chains can be kappa or lambda, and the heavy chain present in IgG is represented as gamma. IgG has 4 subclasses in humans, that are IgG1, IgG2, IgG3, and IgG4, having gamma 1, 2, 3, and 4 respectively. The different IgG subclasses have slightly different structures and functions, although they are closely related to each other. Some important functions of IgG include: it is the only class of immunoglobulin capable of crossing the placenta. Consequently, it provides some degree of immune protection to the developing fetus. Among its subclasses, only IgG3 and IgG4 can cross placenta. The Fc portion of IgG can bind with complementary proteins. Mainly, it can bind avidly with C1q, an important protein that causes complement activation. Of the 4 subclasses, Ig1 and IgG3 activate complement most efficiently and are responsible for clearing most protein antigens. It can also bind to the phagocytic cells once they are attached with antigen, thus making engulfment of that invader easy by phagocytic cells, under a process called opsonization. IgG1 and IgG3 are also efficiently involved in opsonization, thus facilitates the process of phagocytosis. But IgG2 and IgG4 react predominantly with carbohydrate antigens and are relatively poor opsonin. IgG can also attach with Fc receptors present on monocytes, macrophages, and other immune cells and aids in their activity against foreign antigen. Next class of antibody is Immunoglobulin M. IgM is the first antibody produced in response to a microbial attack by B cells. It circulates in the blood and lymph and constitutes 6% of the total antibody content in the serum. Structurally, it consists of a pentamer of 5 immunoglobulin molecules joined together by disulfide bonds, making it a largest antibody among all classes. Here an extra protein chain called as joining or J chain is present, That is attached to the heavy chain of 2 of the 5 immunoglobulin molecules that make up IgM. This J chain is required for the proper polymerization of IgM into pentamers, and in the absence of a J chain, pentamers are not formed. So this pentameric structure provides 10 antigen binding sites to one IgM molecule. Although steric hindrance is there, that practically allows it to use 6 antigen binding sites at once. These large number of antigenic sites facilitates efficient activation of immune system. IgM is extremely efficient at binding and agglutinating microorganisms early in inflammatory response. It also plays role in opsonization, thus helping in the process of phagocytosis. It is also very efficient at making larger immune complexes, which can activate unwanted inflammation and ultimately an inflammatory disease. That's why its production is downregulated as soon as sufficient IgG has been generated. It has 5 complement binding sites that makes it efficient in complement-mediated lysis of the organisms. Next class of immunoglobulin is IgA. It is the major antibody found in body secretions like tears, sweat, saliva, breast milk, serum, contents of lungs, gut, urine, etc. It is also present in mucosal lining of respiratory and intestinal tract, making it a major mucosal immunoglobulin. It constitutes 13% of the total antibody content in the serum and is divided into 2 subclasses that are IgA1 and IgA2. It exists in both monomeric as well as dimeric forms. In dimeric form, it is composed of 2 Ag molecules, 1 joining chain attached with heavy chains of 2 immunoglobulin molecules, and a secretory piece. Attachment of secretory piece prevents digestion of this immunoglobulin in the intestinal and bronchial secretions. IgA2 is the predominant subclass in secretions and neutralizes antigens that enter via these mucosal routes. IgA1, the monomeric IgA in serum, is capable of neutralizing antigens that enter the circulation, but IgA1 is sensitive to bacterial proteases and therefore less useful for host defense at mucosal surfaces. IgA has additional functions via its Fc receptor, present on mononuclear cells and neutrophils, for activation of phagocytosis, inflammatory mediator release, and antibody-dependent cell-mediated cytotoxicity, ADCC. Another class of immunoglobulin is IgD. It is present as a monomer and weighs around 180 kilodalton. It comprises less than 1% of the total antibody content in serum. IgD generally has a low serum concentration and is unstable in serum, being quickly degraded by serum plasmin. IgD is synthesized by antigen-sensitive B lymphocytes, is not secreted, acting as a cell surface receptor for activation of these cells by the specific antigen. It exists as a monomer of the basic Ig structure, but the functional significance of serum IgD is not clear. The last class of immunoglobulin is IgE. IgE is present in the least amounts, around 0.02% of the antibody content in the serum. These are present in the linings of the respiratory and intestinal tracts and respond to allergic reactions. Each molecule of IgE consists of 1 4-chain unit and so has 2 antigen-binding sites, like the IgG molecule. However, each of its H chains has an extra constant domain, CH4, which confers on IgE the special property of binding to the surface of basophils and mast cells. When antigens bind to these attached IgE molecules, the cell is stimulated to release chemicals such as histamines that are involved in allergic reactions. Ig antibodies also help protect against parasitic infections. So this was all. After knowing about different classes of immunoglobulins, next section will help you understand detailed functions of immunoglobulins or antibodies.